evening, everyone. Welcome you all to the online lecture series of Physiological Society of India. Today it is the time for 10th lecture series of PSI. This lecture series of PSI was initiated on 13th July 2023. The objective of this lecture series is to add on the needs of the curricular aspects of physiological courses at both undergraduation and post-graduation level. The Education Committee of PSI regularly organizes this online lecture series on fourth Saturday of every month from 7 to 9 p.m. The theme of today's series is based on the special senses of human body. Like other sessions, today's also we get the opportunity to hear from two honorable speakers, Mr. Prosenjit Choudhury and Dr. Arnab Dash. Welcome Mr. Choudhury and Dr. Dash in today's session on behalf of Physiological Society of India. Today's first session is based on the act of seeing, that is vision, and the act of taste sensation, that is termed as gustation. Prosenjit Choudhury, today's first speaker, will be discussing on this matter. Prosenjit Choudhury passed BSc in Honours in Physiology from Surendranath College under Calcutta University in the year of 2005, followed by MSc in Physiology from Rajabaja Science College, Calcutta University in 2007. After completion of Masters, Choudhury served as government-approved part-time teacher in Sonarpur Mohavidyalaya, West Bengal from 2007 to 2015. Then he joined as assistant professor in West Bengal Education Services in the year of 2015. Since then, he is acting as assistant professor and head of the Department of Physiology in Government General Degree College, Mohanpur, Paschim Medinipur, West Bengal. Choudhury is interested in research on reproductive health of adolescents. Today, he will be discussing on the topic entitled From Seeing to Savoring, Demystifying Vision and Gustation. Welcome, Mr. Choudhury, again. Please, Mr. Choudhury, start your topic. The stage is over to you.
Thank you, Dr. Dash. Am I audible and visible?
Yes, sir. You are audible and visible both.
Thank you. Good
Thank
evening,
you so much.
everyone. Good evening, everyone. Today, I have been given an opportunity to deliver a lecture for undergraduate students on vision and gustation. I hope as we delve into this topic, we will gradually appreciate the intricacies of how our senses are organized. So, let's begin. Uh, I'm sharing my screen. Madam, is it visible?
No, sir. Your screen is not visible. Not yet.
Okay. Now,
No, sir. No.
okay, okay. Is it visible now?
It's coming, I think. Yeah, it's visible,
Is it
but yeah, it's visible. Yes, 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 visible. But make it full screen, sir.
okay? Okay, okay, okay.
Yeah, now it's
Now,
okay, okay. Okay. Yes, it's
okay.
visible, and it's total visible. Okay.
Oh, okay.
Full screen.
Thank you. Thank you.
Thank you.
So we are starting our lecture. Okay. Okay, friends. Do you know why some senses are called special senses? The answer is very easy because these senses are very special, so special that all the senses has their specific organs. Like vision. Vision has its own organ, that is the eyes. Eyes are most romantic organ what we have actually. Through your eyes, you not only see the entire world, but also entire world can see you, can know your can know your emotion, can understand your feelings. So let's see about the this topic, this about this more in more detail. Okay, first of all, we are starting with the anatomy of the eye. You see, eyes are like two spherical structure, like two table tennis ball filled up with fluids. So the as it is a sphere, so in any sphere, the inner part is that which is more closer to the center, and outer part is those which are distant to the center. So the most inner part of eye is our retina, which is the light sensitive neuronal part. This is a retina, the most inner part. This is retina, and it is similar with our if you compare it with the camera, then it is similar with the film or photosensitive cells of digital camera. Okay, so then next part, just adjacent to the retina are three parts which are connected with each other, three distinct parts but connected with each other, collectively known as uval tract. It consists of, first of all, choroid. This is the choroid, this red one, this segment is choroid. So choroid is a rich capillary bed with the light absorbing pigment melatonin. Then the next part is the ciliary body. It is a ciliary body section which actually adjusts the lens. Ciliary body with the zonule fiber, it adjusts the lens for focusing. Okay, the third one is the iris. This is the iris. This iris section is, it control the size of pupil. If you draw a picture of so it is a pupil, and this pupil is required to adjust the amount of light which enter within the eyes. Now the most outer part is called the sclera. Sclera is a tough white fibrous tissue layer. In the frontal part of the eye is called cornea. Cornea is made up with transparent tissues and permit the light to enter within the eye. Now, as we told earlier, the eye is filled up with fluid. So there are two types of fluid. In this area, in the frontal part, there is aqueous humor. This aqueous humor is thin fluid which is like the water, and there are two chambers for aqueous humor. One is the anterior chamber. Anterior chamber is from cornea to lens, and then cornea to lens, that means this area, this part, cornea to lens. And then the posterior chamber, that is the iris to lens. Iris means this area to lens, means that this is the posterior chamber. Now you see aqueous humor actually from the ciliary process. What is the ciliary process? Ciliary process is present here. It is the vascular part of the ciliary body from this area, aqueous humor actually formed, and it is drained through the limbus. Limbus is present in this area at the junction between cornea and sclera. This is the area here limbus is present. If the drainage system is somehow obstructed, then serious problem may arise like glaucoma. Now we will see what glaucoma is or how it failure of drainage through limbus. If the drainage of limbus is failure, then what happened? Then intraocular pressure actually increases, which actually decrease the blood supply to the eye and damage the retinal neurons. As a result, glaucoma develops. That is, that glaucoma actually impairs the vision very much and very common disease also. Then the last one of the anatomy part is the vitreous humor. You can see here vitreous humor constitute the maximum part of the eye, that is the 80% volume of eye consists of vitreous humor. It actually maintains the shape of the eye, as well as it contains some phagocytic cells to remove the blood and other debris. It is very important and very necessary. Why? Because the presence of many debris within this area can impair the amount of blood in amount of amount of light entering within the cornea, which may decrease the acuity of the visual acuity. Okay, then the formation of next segment is the formation of image on the retina. So formation of image in the retina has some factors. There are some factors which actually influence the image formation in retina. The first one is the transmission of light. Light is actually transmitted through two transparent parts. One is cornea, another one is lens. These tissues are very specialized to achieve a level of transparency, and this transparency is very much important to enter light to the retina. But if this transparency decreases or the opacity in the lens increase, then cataract is a very common problem, or cataract is a very common problem, and cataract may develop. You see, over 70 years of age, every individual has a problem of decreasing transparency in their lens, and many of them develops cataract. And we know that modern days cataract needs surgical procedures, phaco or other surgical procedures. And still now 50% of blindness all over the world is due to cataract. So it is very important. Now, another part factor is the focusing of image. So the focusing of image, so image focusing needs the two members are important for focusing an image. Number one is cornea. Cornea actually maximum amount of refraction occur in the cornea. So when light comes, so it needs the refracted, and cornea has the capacity to refract maximum. Its refraction power is even higher than the lenses. Now it can be understood during underwater swimming. During swimming, you may you may observe hazy out of focus images. Okay, it is due to the fact that Unlike air, refractive index of water is similar with the cornea. So in air, the interface of cornea and the air, there is some refraction is possible, but in case of water, the refraction is not at all possible. That's why image do not form on the retina, and we get out of focus image. Next, most important part is the lens. Lens is very much important or most important for focusing due to the fact that refraction of lens can be adjustable. So, lens can focus objects from various distances. It is called actually accommodation. So, it actually achieved through the ciliary muscles. During seeing to the distant object, lens become flat and thin, and when we see the near object, our lens become thicker and rounded. Ah, you see, shape of the lens has depend on two factors. One is the elasticity of the lens that tends to round up the lens. Actually, elasticity of lens actually work in this way. It tends to round up the lens, and if you get the lens externally, or if you pull out your lens from your eyes, you will get the shape is spherical, not elliptical. So you will get the spherical lens. So because the elasticity of lens tend to keep it rounded. On the other hand, the zonule fibers, it tends to make it more flattened. Now, accommodation is the process for focusing near object is that contraction of ciliary muscle, which leads to reduce the tension of zonule fiber, and that increase the elasticity of the lens, and that increase the curvature of the lens. So again, for focusing the near object, contraction of ciliary muscle, which leads to reduce the tension of the zonule fiber, and which may increase the elasticity of lens and then increase the curvature of lens. Now, one of the most important factor is the clarity of image, and which is got by the pupil. So, amount of light is very much important to form an image. So, increased light can tend to blur an image. On the other hand, very low light can decrease the visual acuity. So, exact but accurate pupil size is very important for any image formation. So for the it is got through pupillary reflex, and we will discuss about it later. Now here is the formation of image on the retina. So we got an inverted and smaller image, and this is the optics of the image formation. Human eye and camera. I am skipping the slide. So here are some refractive errors. So you can see here the most common refractive error is the myopia. That is you see, this is a normal shape of the eyeball, and here the eyeball got enlarged. That's why the focusing is not the right place of in the retina. On the in this case, it is difficulty appears in seeing the distant objects, which can be corrected through concave lenses. Another problem is hyperopia, where the eyeball size is reduced, and here you can see it. The focus is beyond the retina, and this problem can be solved with convex lenses. Another very common problem is presbyopia, which is found about all over 40 years of person. Many persons, maximum persons got this kind of problems. That is the difficulty in seeing. That is the difficulty in seeing near objects clearly, and it occurs due to the decrease in the elasticity of the lens. Now, we will see the retina, the most important part of the eye, which is the retina. We will see it in more detail. Okay, so first of all is the histology. So retina is the neural portion of the eye. It is actually a part of the central nervous system. So it is the histology. It is pigment epithelium, which is the most outer part of the retina. It is the most outer part of the retina. Okay. So here is the pigment epithelium, which is adjacent to the choroid layer. Then it is very important for reduced backscattering of light, as well as maintenance of photoreceptor, renewing of photopigments, and phagocytosing the photoreceptor disc, which are very important part for phototransduction procedure. And number two, then we have the 5 types of neurons here. Photoreceptors, which are rod and cone. This is rod and cone. Then the bipolar cell. So it is your bipolar cell. These are the bipolar cells. Then the ganglion cells. These are the 3 types of cells. Another 2 are horizontal cell. This is the horizontal cells. And amacrine cells. These are the amacrine cells. Now, cell bodies are located in inner and outer nuclear layer and ganglion cell layer. So it is a cell body of photoreceptor, rod and cone, that is the outer nuclear layer. The cell body of bipolar cell, inner nuclear layer, and then the cell body of ganglion cell layer. And processes and synaptic complexes are inner and outer plexiform layers. So here is the outer plexiform layer, this is the outer plexiform layer, and this is the inner plexiform layer. And horizontal cells and amacrine cells are very important for lateral interaction in the outer and inner plexiform layer. These can laterally connect more than one rod and cone cell. It is important to process the image and increase the sensitivity. Then the photoreceptors. Rod and cones are photoreceptors. These are the photosensitive cells. So the structure of rod and cone, here the rod outer segment consists of membrane disc. These are disc and these are folds, and cone do not have any disc, it has the folds. This fold and disc actually contains light sensitive photopigments, we will see later. Then the inner segment, this is the inner segment called the cell nucleus. So it is the nucleus part, these are the nucleus, and terminal to bipolar and horizontal cell, that is a synaptic terminals. So this is the structure of photoreceptors. These photoreceptors actually absorb the light by the photopigment, then initiate cascades of events, which is called the transduction event. We will discuss it. Change of the membrane potential, the potential change. Then the change the amount of neurotransmitter for photoreceptors, amount of neurotransmitter from photoreceptors, and ultimately information carried to the brain through bipolar and ganglion cells. Now, the phototransduction mechanism. See, this is a photopigment. Okay. Opsin are the protein part. These are the 7 TM structure, 7 transmembrane structure. It is GPCR, G protein couple receptor, with the 7 transmembrane structure. It is actually attached with a chromophore, which is called the 11-cis retinal. So these 11-cis retinal is the aldehyde of vitamin A. This is the aldehyde form of vitamin A. So when light, so here is the rhodopsin. Rhodopsin is the opsin and the photopigment of rod cell. As rod cell photopigment is actually well experimented, so well informed. So here rhodopsin is shown here. So rhodopsin, when rhodopsin absorbs the light, then 11-cis retinal then converted into all trans retinal. So there is a bending 11-cis, and this become, this is, for example, this is 11-cis and this is the all trans. So conformational change of this chromophore leads to the conformational change of these 7 transmembrane GPCR. Rhodopsin leads to the activation of, and this conformational change is activating this G protein, which is called transducing. Then the activation of an enzyme which is called PDE, that is a phosphodiesterase enzyme, and this phosphodiesterase enzyme is responsible for the closing of sodium ion channel. Actually, what is happening? Say, and that is the decrease of the concentration of sodium ion. Actually, what happened? In the dark, sodium ion actually enters to the rod cell. It enters rod cell and then it release photoreceptor cell release in glutamate and excitatory neurotransmitter, then which actually activates bipolar cell. It releases inhibitory neurotransmitter, and so thus no signal from the ganglion cells. In when light appears to the photoreceptors, then these sodium channels become closed and the entry of sodium ion becomes cyst, and that's why it become hyperpolarized. So in normal time, the RMP is about minus 40 millivolt, and due to the activation through the light, it may vary up to minus 65 millivolt. For your information, there is no action potential found in rod or cone. Thus, a gradual decrease of resting membrane potential actually leads to decrease the release of neurotransmitter, and that actually leads to the signal to the brain. Now, light on a photoreceptor rod and cone change the membrane potential from minus 40 millivolt. This is a progressive increase of potential depending on the amount of light up to 65. No AP, no action potential. Actually, gradual decrease of potential. Ah, Actually, gradual decrease means gradual increase of the potential. Hyperpolarization occurs here. Then the change in the rate of neurotransmitter release from photoreceptors, which photoreceptor which ultimately convey the information to the brain. Here the amplification of the light signals, each cascade absorption of a single photon by rhodopsin molecule can be responsible for the closure of approximately 200 ion channel, which may change the membrane potential up to 1 millivolt. Okay. So here are some functional specialization of rod and cone. Rod are extremely sensitive to light and very low spatial resolution. Resolution is very low, but it is sensitive to light. In low light, rod can be activated. In cone, relatively insensitive to light, very high spatial resolution. Very low level of light, only rod are activated, and this rod activated vision, rod dependent vision is called scotopic vision. In bright condition, that is the indoor lighting or sunlight, rod saturated. Rod cell becomes saturated and only cone are activated. This is called photopic vision. And low level of light like the twilight time, starlight, moonlight, both rod and cone are activated. It is called the mesopic vision. So it is a absolute threshold for rod and cone, of obviously our cone. This is the cone threshold. This is mesopic region, and this is rod saturation begins at that region, and this is the photopic, that is only cone. Activation of rod needs only a single photon, whereas activation of cone need more than 100 photons. So rod is much more sensitive than cone, but whatever image we are seeing, whatever high resolution image is possible only for cone, not for rod. Here is the cone are present. Look, here is the location of rod and cone in the retina. Cones are present at low density throughout the retina. Throughout the retina, you will find cone in very low density with a sharp peak at the center of fovea. This green color is a cone. This is the fovea. Fovea is a central part of retina where maximum light appears. So acuity may be gain here maximum amount. Then rod are present in high density throughout the retina. Rod are present high density. Here you can see rod high density. This is the density of rod with a sharp decline in. Here you can find uh, here you can find the decline of the rod in the fovea region. Now, the cone are responsible for the color vision. Three different type of cones are found responsible for. One is called the S, another one is M and L. Blue or short wavelength, green or medium wavelength, red or long wavelength cone. So these different types of cones, three different types of cones are found, and they are responsible for color vision, they can be stimulated. They are actually stimulated in different segment of wavelengths. So how cone and higher order neurons produce sensation of color is still unclear. So the actual neural mechanism, detailed neural mechanism of color vision is still not understood clearly. And this is the visual pathway. So visual pathway means how does these visual information are sent to the brain. So ganglion cell, as we know from the rod, this is a rod, there is a bipolar cell, then this is the ganglion cell. So ganglion cell action exceeds the retina through optic disc region or blind spot. Blind spots mean from the spot where all the nerves exit from the retina. Then these bundles together, to form optic nerve. So these nerves are actually form a bundle called the optic nerve. Okay. Then they straight goes to the optic chiasm at the base of the dancephalon. So this is the optic chiasm. This is the optic chiasm, the A shape. This X shape, just at the base of dancephalon, is called the optic chiasm. 60% of fiber cross to the opposite side. 60% of fiber is here, you can find 60% of fibers. So if it is a 60% of fiber cross to the opposite side, 40% remain in the same side. So now ganglion cell action on the, and now ganglion cell actions after crossing the optic chiasm segment, they form the optic tract. So here is the optic tract, here is another optic tract. So each optic tract contains neuron from both sides of retina. And this optic tract now supplies to the different structures of dancephalon and midbrain. First, they terminate in lateral geniculate nucleus, dorsal lateral geniculate nucleus of the thalamus, from where they send action, or the action of the they send action to cerebral cortex via internal capsule, and cerebral cortex they terminate in primary visual cortex or striated cortex area, which is the Brodmann area 17 or V1, which is present at the occipital lobe, that is the backside of your head. And number 2, after crossing the optic chiasma segment, this segment, after crossing this segment, optic tract, number 1 first terminate into the LG, lateral geniculate nucleus of thalamus, and some of the fibers terminate in pretectum area. What is the pretectum area? Pretectum area is area between the dancephalon and the midbrain. That means the thalamus and the midbrain area. So this is called a pretectum area. Here is a nucleus called Edinger-Westphal nucleus. It is present just beside the oculomotor nucleus area. Then from Edinger-Westphal nucleus, preganglionic parasympathetic fibers arise, and that send action through oculomotor nerve and terminate on the neurons in the ciliary ganglion. This is the ciliary ganglion. And this is the pretectum area, and this is uh, sorry, this is the tectum area, and this is the Edinger Westphal nucleus. And from here, the preganglionic parasympathetic fibers in cranial nerve 3, and they terminate here ciliary ganglion. Then after ciliary ganglion, the postganglionic fiber terminate in the constrictor muscle of the iris and decrease the diameter of iris. Okay, this is called pupillary reflex. That means when increase the amount of light give rise to the constriction of the pupil. Okay. Other target of ganglion cells are suprachiasmatic nucleus of hypothalamus. Here they can control the biorhythm, that is the circadian rhythm. And another part is superior colliculus. Superior colliculus is the part of the midbrain, tectum area. Uh, it is responsible for head and eye movements. So that's all about the vision. And now we would look what the gustation is. So gustation is to detect the stimuli in the food through the taste buds present in the tongue. So first, innervation of the taste cells. You see, anterior two-third of the tongue, from anterior two-third of the tongue, the chorda tympani and the greater superior petrosal branch of the facial nerve. So anterior two-third of, actually taste buds are present all over the mouth and actually on the tongue, basically on the tongue and the upper part of alimentary canal. Taste buds are found. So taste buds are innervated through different cranial nerves, and the anterior two-third of the tongue is innervated by the facial nerve 7, two-part facial nerves, chorda tympani and the greater superior petrosal branch of facial nerve. Then the posterior one-third of the tongue is innervated through lingual branch of glossopharyngeal nerve, that is the cranial nerve 9, and the epiglottis part is innervated with superior laryngeal branch of vagus nerve, that is the cranial nerve 10. And all of these stimulus actually goes to an area which is called the solitary nucleus of the brainstem. From here, from here, the impulses goes to the VPM, that is the ventral posterior medial nucleus of the thalamus, and from here to the gustatory cortex of the two areas of the cortex, two gustatory cortex. One is present in the temporal lobe, that is the insula part, and another one is present in the frontal lobe, that is the operculum part. So so here the nerve supplies to the insula and the frontal cortex. And this is the path for the primary taste perception and reciprocal projection to the hypothalamus and amygdala. This is also an important part which is responsible for appetite, satiety and other homeostatic responses associated with eating. Now, taste perception in human. So there are different types of taste. Actually, we can taste various type of taste, various, but the primary taste categorized. But primary taste we can find salt, sour, sweet, amino acid or umami and bitter because they have distinct receptors. They have separate receptors, okay, for these sensations. Now, the taste buds. We have 4,000 taste buds throughout the oral cavity and alimentary canals, and the small elevation on the tongue is called the papillae. Taste buds actually present in the papillae, and which are the small elevation on the tongue. There are mainly three types of papillae. One is called fangiform, another one is circumvallate, and the third one is foliate. It is 25%, 50%, and 25%. So fungiform papillae are found anterior two-thirds of tongue. Circumvallate papillae are found in rear of the tongue, and foliate papillae is found in posterolateral tongue. So these are the different taste buds, type of papillae, and these are the taste buds. This is the structure of taste buds, and here are the taste cells. Taste cells, actually taste buds consist of the taste cell and some supportive cells. So in taste cell, there is a apical surface, which apical surface means that this is apical surface. So clustered in a small opening, this is a small opening at the surface of the tongue. It is called taste pore. In the basal surface, that is the basal surface, synapses with the various cranial nerve as discussed earlier. Now the final part is the taste perception in the human. So as we told earlier, all of the taste, there's a primary taste. We have distinct receptors, like salt has amyloride sensitive sodium ion channels, sour has hydrogen sensitive cation channels, sweet has T1R2, TR3, T1R3 receptors, and amino acid has T1R1 and T1R3 receptors, and bitter has T2R receptors. So all primary taste sensations has their distinct receptors, and so thus they have the distinct activity or signal transduction mechanism. So you see salt, and in case of salt and acid, they are the ion channels. This is amyloride sensitive sodium ion channel is a sodium ion channel, which actually when binds with the sodium open up, and that allows the cation to enter. So similarly, hydrogen sensitive cation channel, which allow the cation to enter in case of the acid, that is a sour. The rest of the three, that is that are sweet, amino acid, and bitter, they all are G protein coupled receptors. The receptors are different, that is the T1 or 2 in case of sweet, amino acid. All other receptors are different, as we told here. They all actually stimulate phospholipase C beta 2, phospholipase C beta 2, and here is the G protein. In case of bitter sensation, the G protein is called the gustation. Actually, gustation protein is mostly found in bitter receptor. So when phospholipase C activated, the second messenger IP3 is formed, and IP3 actually opens the calcium ion channel, which is TRPM5 channel. In all cases, the channel is TRPM5 calcium ion channel. So calcium ion enters within the cell. Calcium ion enters within the cell. So calcium concentration of calcium increases within the cells, and which actually help to release the neurotransmitter or activation of depolarization, or the action potential generation, and eventually release of the neurotransmitter. Eventually, neurotransmitter. So, so that's all. Thank you.
Thank you so much, sir, for your very nice presentation. Um, Mr. Chaudhary discussed so many aspects related to gustation and vision.
Thank
He nicely demonstrated the anatomical structure of eye in comparison to digital camera. He also mentioned the histology of the retina. He said about the formation of aqueous and vitreous humor. Sir also mentioned the reason to develop the glaucoma, and how is myopia, presbyopia, etc. Sir very nicely represented how image is formed on the retina and how cataract improve the vision. He said about the photoreceptors, rods and cones, and how they are acting at different light status and how phototransduction is completed. He also stated visual pathway, blind spot, etc. In case of gustation, the organization of the taste system is nicely demonstrated and various types of taste perception in human body. It was a very, very nice presentation, wonderfully sir demonstrated in detail the structure as well as the function related to vision and the gustation. Thank you so much, sir. In part of the audience or participants, if you have any question, please feel free to ask, sir. Or otherwise, you can put your question in the chat box. It will be replied shortly. Thank you so much, sir. Now we are going to the second session of today's PSI lecture series. Uh, in the second session, the speaker is Dr. Arnab Dash. Dr. Arnab Dash completed his bachelor and master's in physiology from Hooghly Mohsin College, Hooghly, West Bengal. He pursued his research work for PhD in collaboration between the University of Burdwan and Sports Authority of India, Bangalore. Presently, Dr. Dash is an assistant professor in the Department of Sports Science and Yoga at Ramakrishna Mission Vivekananda Educational and Research Institute, Belur, Kolkata. He is also heading the exercise physiology and clinical evaluation laboratory there. Along with this, he is associated with Sports Authority of India, Calcutta University, and also associated with Ram College as a guest faculty of exercise and sports physiology. Prior to joining this position, Dr. Dash was the assistant professor in the Department of Exercise and Sports Science at Manipal Academy of Higher Education, Manipal. He worked at Sports Authority of India, Bangalore in the Exercise Physiology Lab as a research fellow, where he worked with several national teams like hockey, rowing, volleyball, basketball, swimming, athletics, and several allied athletes of international repute like Olympics, Commonwealth Games, Asian Games, etc., targeting optimum physical fitness and wellness. He is continuously working with the budding and grassroots level athletes as well to optimize performance and identify talent. He is actively associated with district sports association and sports academics for accomplishing his social commitments towards supporting young players. Till now, he has more than 4 years of experience in teaching postgraduates. So far, Dr. Dash has 11 research publications in international journals. Currently, Dr. Arnab Dash is guiding three PhD scholars and his broad area of research interest in exercise physiology and sports genetics. Today, the second session is based on the system associated with hearing, that is, auditory system, and the system associated with smell sensation, that is, olfaction. Dr. Dash will be discussing on the topic entitled Overview of Spatial Senses, Auditory and Olfactory System. Dr. Das, welcome you again. Please start your session. Thank you so much.
Thank you, Dr. Dash, for this
Thank you,
beautiful,
sir.
nice introduction. Hope I am audible clearly. Yes. Thank you. So I'll quickly share
Thank
the
you, sir.
screen, uh, and please let me know is it visible or not.
Yes, sir, it's visible.
Is it visible?
But yes, sir, visible.
Okay.
Yes. Thank you.
Okay.
Please start.
Thank you. So should I start? Okay. Okay. So first of all, good evening, everyone. And first, I'd like to thank Physiological Society of India for this great initiative of online lecture series. And special thanks to the PSI Education Committee, all the members, for doing it regularly. And today for inviting me to deliver a talk on overview of special senses, particularly emphasizing the auditory and olfactory system. And just now we have finished the vision and the gustation, that is the sense perception. Now what you are hearing, the audition, so that part will focus, and most probably it's almost after some time will be the dinner time. So if something is getting cooked in your kitchen, so most probably the olfactory system also will focus. So the physiology mainly. So, first, a very brief introduction about our ears. And if we see, we have multiple function through our ears. Ears plays a very important role to maintain our balance. And most probably, some of you have faced the that if you are going to a high altitude, for example, if you are going to a Darjeeling, it's not very high altitude, but Darjeeling or some aeroplane exposure or a flight exposure, then most probably some of you have faced that your eardrums or some pressure changes are happening. So ears are also regulate our the pressure changes, atmospheric changes of the pressure, and some certain physiological responses also occurs. And the from the childhood we have heard that the ear also houses the very smallest bone in our body. That's name is stapes. So the question is another will focus in this part is not the balance, not the pressure changes, how the ears is doing, or the about the pure anatomy. Rather, we'll focus only on the hearing part. But before doing starting that, this ears has a very strong effect or role in the childhood punishments as well. I also got. And the outer part of the ears is mainly responsible from the basic pulling to the punishment to the complex punishments as well. An outer part of the ear, that is known as the external ear, also plays an important role in terms of fashion, designing, or look as well, through piercing and through ornaments, particularly, both in females as well as in males nowadays. But our main focus, as I told, will be focusing on hearing. So how we are hearing these words or how our brain is processing, that part we will not enter, but how it's reaching the brain, that words, that we'll see in the next couple of minutes. So if we see the auditory system, and let's start with the anatomy of the ear. The outer part, the known as the external ear, and very in that external ear also, some portion we can see visually, and the inner part of the middle ear or inner ear we can't see visually until and unless through some operation or something is going on. So outer ear mainly basically composed of an auricle, which is also known as pinna, the helix, I'll just open the pointer, and the lobule. As well as it also consists of the auditory canal, which contains certain glands that secrete cerumen. It's basically a wax protecting substances. Now, if we see the different parts of the middle ear, so middle ear consists of a tympanic membrane, the bluish one, you can see the blue color here, the tympanic membrane, also known as eardrum. Most probably if certain infection or some punctures happen, some painful things, and those things can also occur. And the next part is basically known as the ossicles, which composed of three bones known as malleus, incus, and stapes. In that, stapes is the very small, it's the smallest bone in our body. It also contains the eustachian tube and a oval window. Next, coming to the inner ear, it's a very basic anatomy if we go through. So the inner ear consists of labyrinthes and known as labyrinth, and there are two different types of labyrinth. One is known as bony labyrinth that basically contains perilymph, one particular fluid, and membranous labyrinth, which is filled with potassium rich fluid called as endolymph. Basically, the pressure changes in this perilymph and endolymph is responsible to do some changes in that particular portion, which causes us or made us to hear something. So when we are hearing something, obviously through ear the sound is traveling, and it's basically causing changes in the perilymph and endolymph. So that part we'll go through. So the labyrinth has three components. In the inner ear, the first one is the very complex one is the cochlea. It's basically a spiral shaped organ, consists of a 2 3/4 turns from the broad base to the narrow apex that contains the hair cells. Hair cells basically are the receptors which is responsible to produce action potential, or in terms of, it's basically a mechanoreceptor which causes or converts mechanical signal to a electrical signal. That the function of the hair cell or how it works, that will go through in detail after a few slides. It also contains, the labyrinth also contains another component known as vestibule. It consists of utricle and saccule. And in the middle portion here you can see this portion is the vestibule, and the saccule portion and the utricle portion. And it also consists of semicircular canals, which basically contains semicircular ducts. It has three different ducts. One is known as the anterior, second one is the posterior, and the third one is the lateral. So these basically the very brief overview about the external ear, different parts of external ear, different parts of the middle ear, and the inner ear as well. Now we'll move to the neurons, or the different components of the vestibulocochlear nerve. So based on this, here you can see the cross section of a cochlea, where you can clearly see the medial portion and the cochlear different portion. But here we are trying basically focus. Here you can see some nerves. One is known as the saccular nerves. Another one is known as the vestibular ganglia. Another one is the utricular nerve. And the major, most important part is the vestibulocochlear nerve, the eighth nerve, that consists of vestibular branch as well as cochlear branch, as it's composed of both the functions. So vestibular, which mainly responsible for maintaining our balance, and the cochlear part, which is mainly responsible for hearing and audition part. So these two nerves basically originates from these places. So the pathway also we'll see. And when we are talking about cochlea, the organ mainly responsible for this audition mechanism is known as the organ of Corti. An organ of Corti, if we focus in the place, here you can see the spiral organ and the organ of Corti in between. Apart from that, just above the organ of Corti, we have scala vestibuli, another one part in the cochlea which contains the perilymph, another vestibular membrane. Then it starts glial duct that contains endolymph, the composition of perilymph and endolymph, the concentration of sodium, potassium in the perilymph and endolymph is completely different. That basically makes the concentration gradient difference and some voltage difference as well. So that potential is known as the endocochlear potential difference. So that part also we'll discuss after some time. Next is the basilar membrane. This organ of Cortis basement is basically known as the basilar membrane. The movement of this basilar membrane and tectorial membrane is responsible when we are hearing something or some pressure changes are happening, particularly in the perilymph and in the endolymph portion. And at the bottom we have, or in the cochlea rather we can say, the scala tympani. That also contains the perilymph. So perilymphs are at two places. One is the scala vestibuli and another one is at the scala tympani. And the vestibular, the tectorial membrane just above that, the cochlear duct, that basically contains the endolymph. So altogether from the organ of Corti, so the cochleal branch of vestibular nerve originates and it gets transmitted, which we saw just in the last slide, the previous slide. So organ of Corti, when we are talking about the organ of Corti contains certain cells and the very specialized cells which is responsible for hearing, that is known as, there are two types of cells. One is known as inner hair cells and another one is known as the outer hair cells. So here in this picture you can see the tectorial membrane, the outer hair cell or the inner hair cell, the head portion, mainly the outer hair cells portion is attached with the tectorial membrane. So when there is a certain vibration or changes in the pressure in the perilymph or endolymph, that region, that means the pressure changes in the scala vestibuli or scala tympani particularly. So certain these movements of the outer hair cell or inner hair cell occurs, and that movement or the mechanical movement basically gets converted into electrical movement by these two specialized cells. So now you will be surprised, there are around 20,000, in some books it also suggests that 16,000, so around 16 to 20,000 outer hair cells are present in our ears, and around 3,500 inner hair cells are also present in each human cochlea. So it's a huge number which is continuously working, and when particularly in this class or in this webinar, when we are hearing something or your own voice or when you are hearing something, so all that cochlear hair cells are constantly working to formulate that sound and to convert it into electrical signal that produce the action potential and to process that information in our brains accordingly. So now it's we'll quickly go through some basic fundamental principles of sound waves, frequency and intensity. So first, when we are talking about hearing, obviously we are trying to detect or trying to interpret the sound which is coming or getting originated at some certain source. So what is sound basically? So sound is basically produced by the compression and decompression waves in the air or in some other elastic media as well in water. So most probably you have seen or you have faced. So if you went to a swimming and you are in the underwater or some underwater, so from outside, hearing that particular information or sound, or rather I can say stimulus also, or the words, is little bit difficult. Rather, if we are in the air and the source of sound is also from the air. So basically the speed of sound propagates depends on the medium as well. So speed of the sound through air is approximately 340 meter per second and the speed of sound through water is 1500 meter per second. Okay. So what is the compression and decompression of waves means basically? So if we just in our physiology practicals, we used to do some practicals related to the tuning fork, and if there is some kind of vibrating body, in our case it's a vocal cord in our body. So if it vibrates, it produces certain kind of sound. So and that particular sound is nothing but a compression and expansion of air molecules that is producing. In this case, in this picture, you can see through the tuning fork, and in this case or in other cases when we are talking or when we are speaking, in that case the molecule vibration is happening due to the vibration of our vocal cords. So in these sound waves we have certain aspects we need to know, that is the frequency and intensity. So every sound wave is a travels through a rhythm, and that is basically known as a particular one cycle of this particular thing is known as the wavelength. So this wavelength can be a little elongated or it can be shortened, as you can see in the downward picture, which I am pointing out. So where here the wavelength is very short or of a source, the amplitude of the sound can also be lower or higher. So these basically indicates or the amplitude or the frequency of sound basically determines the intensity of that sound or the pitch of that sound or the how difficult or how is it a very soothing sound or the sound whatever is getting generated, it's a very painful for our body or painful to us. So that thing. So now if we see sound intensity is measured basically in the units called decibel. So sound pressure level, that is basically the technical term SPL, sound pressure level, and it gets detected by the unit known as decibel. So in our case, in humans, the threshold, the point at which an average young adult can just distinguish the sound from a completely silent situation, and that situation is known as the or demarcated as the 0 decibel. So for example, if leaf rustling or a very polite sounds is basically it's a 20 decibels sound. In library, it was observed that the decibel is approximately 35 to 40. In case of business offices, it's little can be a little chaos or those things, so can be a 60 to 70 or completely outside. If a heavy truck is going, so that can produce a 90 decibel sound as well. Or some drilling work is going on, kind of a jackhammer that can produce more 100 and 120 decibel sounds. And as the decibel increases, our hearing sensation or the pleasantness gradually decreases and can lead to a situation of pain as well. So for example, the jet takeoff, so that sound is approximately more than 130 decibel, and beyond 140 decibel it's purely can elicit the pain and can leads to some noise pollution or some hearing loss as well if the somebody is exposed to this kind of decibel sounds regularly. So an increase in the decibel presents the tenfold increase in sound intensity. So based on that, so now we are hearing, or obviously in a polite tone, when we are hearing something or when we are speaking to someone, so that is within the decibel limit and most probably approximately 60, 70 within that decibel. And how those particular sound waves is basically what kind of physiological responses occurs, now that we will see one by one. So as in the at the last we discussed about ear cells. So that in the auditory transduction, that is basically converting, as we have already told, it's a mechanoreceptor. So what the hair cell is basically, if we are trying to sense the balance or the auditory stimulus, both the vestibular and auditory system begins with the inner ear and both uses a highly specialized kind of receptors in our cochlea that is present known as the hair cells, and it's purely a mechanoreceptors. That means certain mechanical changes will occur in the cochlea due to certain vibrations that will be induced by the sound wave that traveled, and that mechanoreceptors will convert the mechanical vibrations into electrical signals. That is the main ear cells transduce transaction mechanism. So now, if we see the graphically the anatomy or the structure of the cochlea, so cochlea's base basically can detect the approximately 20,000 hertz sounds. Or as it travels, the cochlear apex part. So obviously, when some sound, it can composed of different intensity and different frequencies. So not that doesn't mean that a particular part of the cochlea will detect all the sounds. So based on the nature of the sound, a different hair cells present in the different parts of the cochlea will basically detect that one. And at the apex, it's approximately 2000 to. So here you can see the high frequency waves. If the sound contains a high frequency waves, approximately 1500 to 20,000 hertz, then the base of the particularly the cochlea will be sense that one. In case of medium frequency waves, approximately from 6000 to 1500 hertz, So the middle portion of the cochlea will basically detect that one, or where the hair cells responsible there, or if it's a very low frequency waves, then the apex of the cochlea will try to interpret those sounds. Okay, so when we're talking about the hair cells, obviously, and the mechanical changes are happening. What happens? The hair cells and the organ of Corti, basically the two parts, the tectorial membrane and the basilar membrane. So the bottom, the basilar membrane basically changes or displaces from its resting position when there is certain kind of sound waves or some sound intensity or based on the frequency. So these basilar membrane position change causes certain changes in the hair cells head, that is known as the stereocilia. So the hairs, the heads present here, the pointer, those hairs are basically known as the stereocilia. So there are certain changes will happen at that particular position. So here through one GIF we'll try to see the mechanism of hearing now. So if we see the mechanism of hearing, so how the mechanism of hearing happens? Sound waves, basically when we are hearing something, the sound waves through the canal or through the auditory canal, it reaches the tympanic membrane. And tympanic membrane is known as basically the eardrum. So the tympanic membrane, certain vibrations will happen at the tympanic membrane, and that alteration in waves of high and low pressure in the air causes that tympanic membrane to vibrate back and forth, as you can see in the GIF. So central area of the tympanic membrane basically connects, that is the middle ear portion, the malleus, which vibrates along with the tympanic membrane. So here you can see that with the tympanic membrane, the malleus bone is attached. When there is a vibration, that mechanical vibration will cause the displacement of the malleus or certain changes in the configuration. So as malleus, incus, stapes, these three bones are connected each other. So the vibration is transmitted from the malleus to incus and then to the stapes, this bone. And these stapes is attached with the oval window. So wherever there is some kind of hammering sensation, so that also basically vibrates the oval window as well. So there is a fight between the inner part and the inner ear and the middle ear as well. So that oval window is a part of a middle ear or inner ear. But oval window, if we consider it as a middle ear portion anatomically, so oval window gets either inward or outward movement continuously happens. And that movement of oval window sets up the fluid pressure, because there certain fluid pressure is there. So fluid pressure waves in the perilymph of the cochlea. So within the cochlea, we have perilymph, that is a particular fluid. So once there is a change in the vibration in the stapes, induced oval window position, either inward or outward, that also increases the pressure or decreases the pressure in the cochleal as well. And as the oval window bulges inward, if there is a pressure from the cochlea, from the stapes, the oval window will bulge inward and it pushes the perilymph to the scala vestibuli region. And that particular scala vestibuli then into the endolymph causes the basilar membrane to vibrate. And this vibration of the basilar membrane will be from the resting level, it can go upward and it can change its position compared to rest. And that will basically cause change in the hair cells positions or the configuration as well. So what kind of structural or the mechanical changes will happen, or the positional change will happen in the hair cells? So the appropriate stimulus for the hair cell is the bending of its hairs, basically. As you can see in this GIF, it basically, the hair cells, certain gates are there, the potassium gates. So the change in those particular hairs, that is the stereovilli, or sometimes also known as the stereocilia. So certain changes in those hairs basically causes influx of potassium from endolymph. Endolymph is basically consist of a high potassium rich fluid. So from the endolymph, it will enter, those potassiums will enter into the hair cells. So the longer stereovilea, so the movement. So now the question is from which direction or which side movement will cause the depolarization of the hair cells. It is towards the longer stereovilea side, or the movement towards shorter stereovilea side will excite the cell. It's actually when there is a changes in the basilar membrane, so the bending of the hair cells will move towards the longer stereovilea, and if the hair cells or the stereovilli are going towards the longer stereovilli, as you can see in the pictures and in the GIF as well, that will excite the cell and will cause the depolarization. How? Because the positively charged K plus, when it is entering the cell, so generally the hair cell potential is approximately minus 60 millivolt. It's that is. So there are different types of hair cells. So here we are talking about the hair cells responsible for hearing. Another in the vestibular cells, vestibular apparatus, which is also inner part of the inner ear. So there are two different cells are also there, that is known as the type 1 hair cells and type 2 hair cells. But here we're talking about the hair cells that is responsible, and the potential of this particular hair is approximately minus 60 millivolt, and the outside endolymph can be a plus 85 as well. So those things we'll see. So that particular change or entrance of the K plus into the hair cells will cause depolarization of the particular hair cell. And once the cell gets depolarized, what will happen? Once the action potential or the depolarization happens and the threshold level crosses action potential elicited, so what will happen? The calcium channels will be opened, and there will be an influx of calcium from the perilymph to the inside the hair cells. And these influx or entrance of the calcium, we all know that the vesicle docking requires the vesicles filled with neurotransmitter used to be present in the hair cells. So those neurotransmitter filled vesicles in the presence of calcium will dock with the hair cell base, and that particular in that place there will be a different afferent neurons. So the afferent neurons axon will receive that, but based on that neurotransmitter release, that synapse or in that synapse release of the neurotransmitter in the synapse will stimulate the next or the adjacent afferent neuron. So that is how the signal transmission occurs. So the mechanical change got converted into electrical signal. Now if we see the negative mechanical deformation, as you can see in the right, so mechanical deformation means away from the kinocilium causes the K plus channels to close. So there is a opposite direction movement of the stereocilius or the kinocilium, and it will cause the closure of those potassium channels. And once these channels are closed, these gates are closed, so there will be no influx of the potassium, and ultimately the cell will undergo hyperpolarization. And at that stage, there will be no influx of the Kf calcium, and there will be no release of the neurotransmitter that will stimulate the afferent neurons. Okay. So now the question is, what kind of neurotransmitter gets released? Mainly the synaptic vesicle that will fuse, and the hair cells will release the glutamate. And that excitatory neurotransmitter will trigger the afferent neurons, and that afferent neuron action potential generation in that particular afferent neuron will relay the auditory signal to the brainstem. Okay, so brainstem to the higher brain center as well. So now, as we have seen that more and more number of K plus is entering into the hair cells. Now the question is, what will happen to this particular potassium K plus if continuously K plus influx is happening in the hair cell? So the cell will be full with lot of K plus. Now that K plus, if the cell releases the K plus through gates to the perilymph where the potassium is low, now obviously after certain time the perilymph also will be reached or full with the K plus, and the voltage in the perilymph will change ultimately. So that K plus needs to be recycled or repositioned. So that is how the endocochleal potential now we are going to discuss, that the resting membrane potential of the hair cells is about minus 60 millivolt. And if you see the scala vestibule in this picture, sodium used to be in the scala vestibule is approximately around 150, potassium 5, and whereas chlorine 125. So where there is some structural or the mechanical changes in the hair cell, the potassium will enter, and ultimately the scala tympani region where the perilimbs are present again. So there the potassium will be reached, and it will release the potassium. Sorry, the potassium will be released into the spiral ligament and the stria vascularis region, and through those particular areas the K plus will again enter into the perilymph. So that is how the potassium recycling is continuously going on, and this particular endolymph is formed by the stadia vascularis, and that is why the endolymph used to have a very high concentration of K plus, because the potassium from where it's getting produced, it's basically coming from the stadia vascularis. And if you see it in detail, so basically here on the right side, you can see this is taken from the Boron and Bolpep. So the perilymph, you can see the perilymph, the 0 millivolt and the K plus concentration is very less. So when it is the function once the fibrocytes accumulate potassium for transfer to the intermediate cells, and when it is reaching the intermediate cell, K plus channels in the intermediate cells will basically generate the endocochleal potential. How? The potassium transferred to the intermediate cell from the perilymph, it's basically will enter into the endolymph, and as a result, endolymph will have a very high positive charge, that is the plus 80 millivolt, and can the concentration of the potassium will be approximately 150 millimolar. So that is how these basically causes the concentration gradient or the voltage difference, the endocochleal potential from 0 millivolt in the perilymph, and in the endolymph it's approximately plus 80 millivolt. Sometimes you can see the plus 85 as well. So scala media is electrically positive by plus 85 millivolt and relative to the scala vestibuli and scala tympani. So compared to the scala vestibuli and scala tympani, the endolymph, the scala media that contains basically the endolymphs, so those are the 85. So positively charged used to be there, and the gradient is approximately plus 85. So at one side is 0 millivolt to plus 85. That is the from between the endolymph and perilymph. And if we see the hair cells resting membrane potential, that is approximately minus 60 millivolts. So when there is certain depolarization, the resting membrane potential will change and the action potential will be developed, is to get developed in the hair cells. So this is how the mechanical sensation or the mechanical function got changed into an electrical one. And that is why when we are talking to each other, or when we are continuously speaking or listening to music, or some good notes are coming or some bads also, so all the things the hair cells are basically processing, and that is through the whatever the sound waves or the air pressure that is triggering or the changing the vibration in the in perilymph and endolymph and perilymph portion, and that basically changes the basilar membrane position, and that is how this kind of lot of things are continuously working in our ears. And if we sleep also, most probably we will be surprised, when we are sleeping, still the ears, basically our ears keeps on working continuously. Some sounds most probably the brain will not process, or it will not focus that much, rather will not do proper action, but it's continuously work, it continuously works, the ears basically whatever the sound. If there is a silence also, after that if there is some sound during sleep also, our entire ears, both the ears keeps continuously working. So now if this kind of changes are happening, obviously the auditory information, this action needs to travel from the cochleal branch of vestibulocochlear nerve, and it needs to process to the brain. Otherwise, how we'll interpret that particular information or to memorize that information? Every time it requires to reach the brain as well. So auditory information is conveyed along the cochlear branch of vestibulocochlear nerve, and then it reaches or crosses the brainstem. So there is a cochlear nuclei in the medulla oblongata. So from both sides of the cochlea, the neurons, vestibulocochlear nerve process reaches the middle oblongata, the part of the brainstem. We all know that brainstem consists of a midbrain, pons, and medulla. So first it will enter through the middle oblongata, and after that it will reach the superior olivary nucleus in pons. So once it reaches the superior olivary nucleus in the pons through lateral menisci, it will reach the medial geniculate nucleus in thalamus. It will reach the thalamus, and from the thalamus, that another one neuron will generate, and it will send the signal to the primary auditory cortex in the auditory area in the cerebral cortex. So this is how the auditory information, which was just a vibration or the pressure changes in the air, that got converted into electrical and it reached the brain. It's a very complex mechanism and which is continuously working in our body. So now, if we see, so impairment in this any area, either in the tympanic membrane of the eardrum or in the middle ear or at the part of cochlear level, it can cause, or the exposure, what kind of sound wave exposure, in what physiology or in the factories, there are a lot of sounds, noise, and those things. So based on that, a lot of precautionary measures used to get taken. But now we'll discuss about certain common auditory disorders, and a few of them are hearing loss can be there, tinnitus, a continuous sound that actually doesn't exist from the external source, but there will be a continuous sound or the perception of sound in our ears, or that will brain will ultimately sense. So those particular situation is known as the tinnitus, and certain balance disorders as well can be there. So auditory disorders can have a severe impact in our daily life. What kind of? If there is some auditory disorders, it can affect our working memory, it can affect our attention as well as speech production. It can also hamper the sensory to motor signal relay. That means what we are hearing, and based on that, what kind of actions we are taking. So maybe the command processing also will be impaired. And so comprehensiveness or speech sound information or integration in all those information, those words will be slowly deprived, or the person who is having this particular hearing loss will be very difficult for them to attain those cognitive abilities basically. So now we'll see basically the move to the olfactory system. So now we saw the how the auditory system works. So when we are some sounds, how it get converted into electrical signal, and how the electrical signal from hair cells, the potential changes are happening at the hair cell, and ultimately that information is got relayed to the higher brain center. Now, if we see the olfactory system, and obviously from the image only you can see that we are going to talk about the smell sensation. Okay. So olfaction, or the sense of smell, if we see, smell is detected by approximately 10 million to 20 million receptor cells, which are present in our upper nasal passage. And that is the olfactory membrane of the upper nasal passage. So either can be a good smell, bad smell, or whatever the order is. So that is basically detected by millions of receptor cells present in our nose. Although it looks very small area, but there are millions of receptor cells, and those receptor cells is nothing but a end of the dendrons of certain neurons that we'll see now. So it's estimated that humans can recognize about 10,000 different odors. Obviously, after certain odors, or if multiple odors we are smelling together, maybe we will not be able to recognize or distinguish with each other, and some complex mechanism also happens there. But we can recognize about 10,000 different odors. And the Professor Richard, Dr. Richard Axel and Linda Bach won the 2004 Nobel Prize for their discoveries of this odorant receptor. They got awarded in 2004, but their work was around 1990s, so that period. So the odorant receptor there, and the signaling pathway and the organization of the olfactory system. through the genetic mechanism also they have explained. So for that they got the Nobel Prize in medicine, physiology and medicine. So these olfactory receptors are the first order neurons of the olfactory pathway. Unlike the auditory system which we just saw, there are certain cells are there and the neuron, the first order neuron starts based on the hair cells neurotransmitter release. But in case of olfactory receptors, these are purely the neurons and the dendrons that will basically detect the odorant molecules or the molecules which we can smell. So these olfactory receptors is a bipolar neuron. So as I think we all know, I am not going to deeper of the bipolar neuron. So the bipolar neuron with an exposed knob-shaped dendrites and an axon projecting through the sebriform plate and ending in the olfactory bulb. So basically, from the opening of the from our nasal passage, the odorant molecule, when some odorant molecule is entering, so it will ultimately sensed by the dendrites present in those olfactory receptor dendrites, and that sense will go to the olfactory bulb and ultimately it will reach to the higher brain center. So now, if we see where the olfactory epithelium is located, it's not at the very beginning or the outer part of the nasal cavity. It's a little bit upward, and the olfactory epithelium there mainly the a lot of olfactory receptors are located. Okay. So superior nasal cochlea, this region, and frontal lobe of cerebellum, it will go olfactory tract from where the olfactory receptor and the neurons will be originated and it will reach. So if you see the right side picture, so this is how the olfactory hair cells. So here you can see the odorant molecules basically get sensed by the olfactory hairs, and this is nothing but a dendrites. Dendrites of what? Dendrite of the olfactory receptors. If you can track my sensor or the thumb, so basically the hand, so olfactory receptors, the bluish in color, having a cell body inside. So these olfactory receptors will basically the olfactory molecule or the odorant molecule will basically bind with that we commonly known as the smell. How? It's basically nothing but a composition of a multiple odorant molecules. So those chemicals or molecules will bind with the receptor and ultimately it will trigger or it will elicit the action potential in those olfactory receptors. And these signal will ultimately go through some the cerebriform plate and it will reach the olfactory bulb. Okay, so from where again multiple neurons will generate and that will reach the send the signal to the next level. So now if we see the olfactory transduction and if we just as a recap, if we see the receptor cilia, it has a very G protein coupled receptor mechanism. What is G protein coupled receptor? Obviously, you know that it's a secondary messenger system or the signaling path. That olfactory receptor, there are multiple types of olfactory proteins or the receptors present in our nose, basically. And based on the odorant molecules, it will bind with the receptor and it will activate the G protein pathway, G protein coupled receptor pathway, which basically causes the conversion or production of cyclic AMP from ATP in the presence of or the activation of adenylyl cyclase by the activity of adenylyl cyclase. And this cyclic AMP acts on the sodium channel and that will open the sodium channel through which sodium will enter into the particular receptor cell, particular neurons or the first neuron we can say or the olfactory receptor rather, and sodium influx causes depolarizing the and it will generate the action potential in the olfactory receptor. Unlike in auditory system where the hair cell, the potassium was causing or was generating the depolarization or the action potential in the hair cells, and here we can see that olfactory transduction sodium is the mainly responsible, that sodium will cause the depolarization and depolarization of olfactory receptor cell membrane triggers ultimately the action potential, and that action potential will be a graded receptor potential and it will ultimately spike and there will be a certain no receptor signals and multiple senses can generate through the based on the odorant molecules and how many times it's getting stimulated. So the olfactory pathway, each side of the nose, we have about 40 bundles of the cylinders, and these neurons are basically the unmyelinated axons of the olfactory receptors, which extend through about 20 olfactory foramina in the cerebriform plate of the endomoid bone. So here in this picture, you can clearly see, so based on what kind of receptor, olfactory receptor is sensing or gets activated, based on that it will reach the, or the sensation or the signal will go to the olfactory glomerulus, the OG, olfactory glomerulus. So either green, orange, so there are multiple, there is to be multiple olfactory glomerulus where the signal reaches. And from olfactory glomerulus through multiple neuronal network, actually the signal reaches to the olfactory cortex. There are involvement of granule cells, there are involvement of mitral cells or periglomerular cells as well. And all these cells basically causes certain release of neurotransmitter, either can be a inhibited or can be an excitatory one. So in this picture, the solid black arrows, basically you can see that inhibits or by the GABA release, that releases the neurotransmitter GABA, and the white arrows in this picture you can see wherever. So these basically are the excitatory neurotransmitter and basically releases the glutamate. So that is how the stimulation or inhibition based on the what kind of sources or the olfaction or the smell is coming, or the odorant molecule bodies entered the nose or the nasal cavity. Based on that it will be sensed and ultimately it will reach the olfactory cortex. So now once the, what is the pathway or how the, or it will reach the auditory cortex, that is the next question. So now we'll discuss about the auditory, olfactory pathway. So olfactory pathway, basically once the olfactory receptor sense or gets activated through G protein coupled receptor and the depolarization, and the receptor will send the signal to the olfactory bulb, and through the olfactory tract, the olfactory bulb targets piriform cortex or olfactory tubercle or amygdala or endothenal cortex as well. So based on that, through those areas, either the orbitofrontal cortex will be activated or the information will reach to the thalamus, hypothalamus or hippocampal formation as well. So if there is a conscious discrimination of odors, that this is a good smell or bad smell, or some, this is a rose flower smell or some kind of another flower smells, maybe marigold or something else. So conscious discrimination of the odor is depend or something else is getting cooked in the kitchen. So those discriminations can be clearly seen or depend on the pathway of the orbitofrontal cortex. So that pathway is mainly responsible for conscious discrimination of odors. The pathway to the amygdala basically mediates the emotional responses to the olfactory stimuli. Based on that odorant molecule, it reaches to the cortex and ultimately will feel or will tell that wow or yuck. Those kind of sensation will come. Oh, it's not a good smell, or it is a bad smell. And the immediate response will be the motor response, that will close our breath or we'll try to hold our breath for some time, or we'll close our try to close our nose, basically the nasal passage, so that it should not enter if it's a bad smell. So the pathway to the amygdala mediates the emotional responses to the olfactory stimuli. Olfactory stimuli is nothing but what kind of odorant molecule it entered and it stimulated. And the last one is the pathway to the endothenal cortex is concerned with the olfactory memories. So as we ages, so this kind of memories is getting continuously stored, that this is the smell of this particular one, that is the another one. So those kind of memories building, the pathway is responsible, that is the endothenal cortex pathway that is concerned with the olfactory memories. So now we'll quickly see, till now we have seen how the olfactory molecule molecularly stimulates the or triggers the signal or causes depolarization of the olfactory receptors, and through from the olfactory receptor after getting activated, how that signal reaches the different areas in our brain. So now obviously we'll see the factors that influences olfaction. And there are certain interesting facts. One disease also we'll discuss, that is the women often have a keener sense of smell than men do, especially at the time of ovulation. The sensation is much greater or sensitive in case of women than men. And smoking seriously impairs the sense of smell in the short term and may cause long-term damage of the olfactory receptors, because during the inhalation, that hot and polluted air can regularly trigger certain sensations, certain olfactory receptors, and slowly the olfactory receptors may get damaged or the entire pathway may get hampered. So causes long-term damage to the olfactory receptors. And with aging also, the smell deteriorates as we ages. And one particular disease that is known as dipsomia, so it's the reduced ability to smell. And during the corona time, I think some of us or most of us has faces that the immediate change was, or the effect, another one effect was on the, apart from the nose and the smell sensation. So we are unable to get or interpret the odor, or maybe that all obviously because of the olfactory pathway and the olfactory receptors were not able to process that discrimination or differentiation, or able to trigger that particular action potential in those cells. So that affected the half of those, but irrespective of that particular disease condition or that virus. So here we can see that aging also causes, affects that our olfactory mechanism, and those over the age of 65 and 75% of those over the age of 80 can reduce the ability to smell, and can also caused by the neurological changes, certain such as head injuries, Alzheimer's disease, can be a Parkinson disease, can also affect certain drugs, certain kind of analgesics or steroid, or just now we discussed the damaging effect of smoking can also influence the olfactory mechanism. So this is all about the just a brief or some light on the olfactory system. So I would like to thank everyone for your patience hearing, and if you have some questions, you can ask. I hope it was an interrupted network interruption was not there. Thank you, everyone.
Thank you so much. Thank you so much, sir, for your very, very nice presentation. We enjoyed a lot this session. Um, in the first session of his talk, Dr. Das has said about the audition. That is, Dr. Das has said how ear is important for various purposes, from punishment to styling. But the most important factor is that the most important function of ear is the hearing. He said about the mechanism of hearing very well as the structure of the ear also, organ of Coti, auditory transduction, mechanism of hearing, and how the ions are involved in this mechanism. Even when we're sleeping, specifically ear is still working. He mentioned that disorders like hearing loss, tinnitus, and balance disorders. These auditory disorders put severe difficulty in our daily life. And in case of the second part of his session, Dr. Das has said about the olfactory system. He mentioned the story of discoveries of odorant receptors, olfactory transduction, and ions involved in the mechanism of olfaction. He demonstrated the olfactory pathway. Sir said how we can discriminate the odors consciously, and accordingly we respond very emotionally to that. And women are have a keener sense of smell than the male persons, and with age the smell sensation deteriorates. And he also said that certain diseases like Alzheimer's disease, Parkinson's disease, and many kind of drugs also deteriorates the sense of the olfaction. That is the sense of the smell actually. So from this session, I think from this session that is we're getting the various type of special senses in the human body, like the olfaction, audition, vision, gustation, and I think it will be very much helpful for the physiology learner, both in the undergraduate and the postgraduate level. Thank you very much, sir, again. in part of the audience or in part of the participants, if you have any question, please feel free to ask, sir. It will be answered shortly. Or if you have any queries, you can ask later also in the chat box, or you can mail also. Now, I would like to request Dr. Gargi Raichudhuri, Madam, the coordinator of today's online lecture series, to deliver the vote of thanks. Actually, Dr. Gargi Raichudhuri, now acting as professor of Nopene Institute of Healthcare Studies. It is a center for postgraduate course in Physiotherapy College affiliated to West Bengal University of Health Sciences. Please, Madam.
Good evening, all. Thank you, Shutapa.
Thank you, Madam.
I, Dr. Gargira Chaudhuri, would like to thank Dr. Prashanpriya Nayak, President, PSI, and Dr. Kaushik Bharati, Vice President, PSI, for taking such a noble initiative. My wholehearted thanks to today's speakers, Mr. Prashanjit Chaudhuri and Dr. Arnab Das, for their well-depicted lectures. I'm really very much impressed by the way they have described, they have explained. It was really, what should I say, really pleasure to my eye and to my ear also. Brilliant. I also thank Dr. Shampa Sarkar, convener, PSI, for our constant support, along with Dr. Vinay Chitturi for their technical support. My special thanks to Dr. Shutapa Das for introducing the speakers and summarizing the lectures in a very well manner. And my thanks to all the respected faculty members for their gracious presence in this session. And last but not the least, my blessings, my thanks to all my students who have participated in this webinar. Now, let me announce the topic and speakers for the next month webinar. Dr. Amarnath Sharma, Assistant Professor, Tombo Riba Institute of Health and Medical Sciences, Arunachal Pradesh. He will be giving presentation on overview of cellular physiology in medical physiology. And our next speaker is Dr. Govindraj Yankham, Professor, Regional Institute of Medical Sciences, Manipur. He will be sharing his thoughts on cell membrane and membrane transport. So thank you once again. It's a very good evening spent today, and hope we will continue this with a flying victory. Thank you so much.
Thank you so much, Madam. So now I think the session is going to be end here. Thank you so much.
Oh, thank you.